Hello class, I hope everyone is doing good and they are in good health. In this video lecture we are going to discuss the remaining two different type of sexual reproduction that is found in bacteria. Okay, so particularly we are going to discuss transduction, conjugation, and then there is a special case that is the case of HFR strain, that is high frequency strain, or sexduction in this video. Okay. So let us begin this lecture with transduction. Well, first of all, what is transduction? Okay, who discovered it? So when it comes to discovery, we see that transduction was discovered by two scientists, that is uh, Gender and Liederberg. Okay, Gender and Liederberg in 1952 in Salmonella typhoidea. Salmonella typhimurium. Okay, so this is a type of bacteria in which, first of all, in 1952, Ginder and Liederberg reported this unique phenomena called transduction. Okay, now what is transduction? Let us see. Now, transduction, it is a process of transfer of genetic material from one bacterium to another by the help of viruses. Okay, or in particular bacteriophage. Okay, so what is transduction? It is a process in which the genetic material, that is DNA, of one bacterium is transferred to the another bacterium by a virus. Okay, now this virus is bacteriophage. Okay, now what is bacteriophage? Let us first understand bacteriophage. Okay, so we can split this term in these two words, bacterio and phage. Right, now you can see that the word bacterio stands for obviously bacteria. Okay, this is for bacteria. And phage, wherever there is this phage, word phage, it means virus. So what does it mean? This means bacteria virus, right? Or this means a class of viruses that infect bacteria. Okay, so bacteriophages are actually the class of viruses that infect bacteria. Okay, there are many members of bacteriophage family. Okay, now how they look like, let us see. Okay, how they appear. So if you see a typical bacteriophage like lambda phage, this is how they look like. They have these hexagonal head, and after this head there is this neck, and then there is a basal plate. On this basal plate there are 6 to 8 tails. Okay, now these tails help in attaching or help in attachment of this virus on the surface of bacteria. Okay, what I have drawn so far is either made up of protein or protein plus uh, carbohydrate, okay, or maybe lipid in some cases. This is outer covering called capsid. Okay. Now inside capsid there is genetic material. So this is the genetic material which I have drawn here. Now here in case of bacteriophage, usually this genetic material is sorry, DNA, and as I said, this outer layer is the capsid. Okay, so mostly the viruses have a genetic material and a capsid. That's it. They do not have cellular organization, and hence they do not show any kind of metabolism, and that's why we consider them as dead outside a host. Once inside a host, they do different things. They hijack the cellular machinery and start showing reproduction. Okay, they show metabolism of their type or their characteristic metabolic activities. You know? Hence, we consider the viruses living inside the host but dead outside the host. So it can either be living or dead depending on the environment. That is, it is neither living nor dead, and hence it is the connecting link between living and non-living. Okay. Now, uh, not only this, the viruses uh, are very unique. Unique as in they play a lot of role in evolution as well. They do the horizontal gene transfer, and whatever we are going to study in transduction, in fact, it is one of the method of horizontal gene transfer. Okay, what is it? You will be better able to appreciate when we will be studying genetics. Okay, until we are doing genetics, it's okay. Once we reach in genetics, I will again emphasize this word. What is horizontal gene transfer? Okay, great. So capsid DNA, this is how a typical bacteriophage look like. What happens? Once there's a bacteria, so let me draw a bacteria here for you. So suppose this is a bacterium. Okay, some problem. Okay. So here, let me draw this bacteria. So this is a bacterium, and this is its genetic material. Okay. So obviously the genetic material is circular. So now it is circular, double-stranded circular DNA is the genetic material in bacteria. And then obviously the cellular machinery, there will be ribosomes and things like that. We are not going to draw it here. Now, the size of the bacteria is typically in microns, few microns, like 1 micron to say, for example, 5 micron, right? But the virus is incredibly small. They are in the range of like 20 nanometers, 50 nanometers, or even smaller than that. Okay. So let me take this virus, okay, and shrink it. Okay? So this is a very small virus, right? Viruses are generally very small. So this bacteriophage is very small as compared to the bacteria that it infects. It sits on it with the help of these tails. Okay. Now let me zoom in to better understand. So this is the bacteria. Now what happens is once this bacteria attaches itself on the bacterium, it okay. Let me redraw it. It starts to inject its genetic material. Okay. It starts to inject its genetic material. Okay. So it will inject its genetic material through this thing into the bacterial cell. Okay, so finally this comes here and then it comes here. Okay, so I am deleting this one. And only this red part is here, right? Now what happens? The bacterial genome is cut at one place. Okay, so let me do it. So say for example it is cut at this place, so the bacterial genome will open up. Okay, and it will, the viral genome will get incorporated into the bacterial genome. Let me do it. So suppose this is the viral genome. Okay, and then the viral genome will get incorporated into the bacterial genome. Can you see? So now what happened is the bacterial genome, the viral genome is injected into the bacterial cell, and then inside the bacterial cell the viral genome goes and gets embedded into the bacterial chromosomal DNA. This process is called integration. Okay, that is the viral DNA got integrated into the bacterial DNA. Great. Then what happens? Let us see. Can you see this whole picture now? Great. This is what happens. Then what happens is after some time the viral DNA inside the bacterial cell starts to make its own proteins. Okay, because this viral DNA has the genes for its own protein. So using that information, using that gene, the virus starts to make its own proteins. At the same time, what it does, is it makes a lot of copies of viral DNA. Okay, plus there is the viral capsid. These things are formed. Okay, many of these are formed inside the bacterial cell. Many of it are formed. Then what happens? This is packed into the virus. Now the crucial step is here. What happens many a times that when this new viral DNA is formed, this has to be split from this side, right? Precisely from this side. This is what has to be done, correct? But because of some errors, many a times, what happens that it gets splitted at this end, okay, or maybe this end. As a result, what happens? The new viral DNAs have some piece of bacterial chromosome. Okay, great. And then these are packed into the bacteria, into the viral particles. As a result, okay, as a result, let me drag it a little bit and we'll see here. As a result, when this new virus particle is formed, this will have a viral DNA with some portion of bacterial DNA. Okay, can you see it? Great. Now consider a situation that this particular virus goes and infect another bacteria. Okay, so say for example here I am taking this new bacteria in blue. Okay, and okay, here I'm taking another bacteria in blue, and this is its genome. Okay, now this virus came, infected this particular bacteria, and again the same thing happened. The uh, viral gene is incorporated into this bacterial gene, and now because of this integration, this bacterial genome looks something like this. Can you see this part? Correct. Now again, this whole process will happen. There will be again splicing. Now this time the splicing somehow is correctly done, and only the viral portion is removed. The bacterial portion remains into the bacteria, correct? And the viral gene is ejected and packed into a capsule. Okay, so again, new virus. What we can see here is during this whole process, at least 2 infections were required, 2 bacterial infections, and in due course what happened, the gene from this bacteria, say A, is transferred to bacteria called B, right? Can you see now the definition? The transfer of genetic material from one bacterium to another with the help of virus, that has happened. So the genetic material from bacterium A is transferred to bacteria B with the help of this bacteriophages. And this is what happens in case of transduction. Okay, great. I hope you understood this whole concept. Okay, and this was reported by whom? By Ginder and Liederberg in 1952 in the case of Salmonella typhimurium. Great. This is transduction. So this we have seen. Let's move to the another type of sexual reproduction, that is conjugation. Okay, let us see what is conjugation. Or before that, let us see who discovered it. First thing, who discovered it? So it was discovered by Liederberg. Okay, and Tatum. Liederberg and Tatum in 1946, a year before our independence, year of independence. Okay, in E. coli bacteria. Okay, now I hope all of you know about E. coli. The full form is Escherichia coli. It is a friendly bacteria to our gut. So all of us have this bacteria in our alimentary canal, mainly intestine, and it is a friendly bacteria. We want it to be there. Okay, it helps in our digestion, it helps in vitamin synthesis. Okay, very well. So in E. coli, Liederberg and Tatum first of all reported the process of conjugation. Now what is that? We have already talked in brief about conjugation when we were studying plasmids, particularly, sorry, plasmids. Yeah, plasmids, as well as when we were talking about pili. Okay, so when we were talking about sex pili or longer pili, then also we spoke of conjugation. At the same time, when we were talking about the F plasmid or F factor, then also we spoke of conjugation. Correct? Let us see how it happens. So suppose this is a bacterium. Okay, this is a bacterium and this is its genetic material. Okay, great. Now apart from this genetic material, this bacterium has an extra pair of genetic material, which is again double-stranded circular DNA, and this is called F factor. It is a type of plasmid. Okay, F plasmid. Now because of this plasmid, this bacteria is called F positive or male or donor. Okay, why? Because on this plasmid there's a gene responsible for making longer pili or sex pili. Okay, so this structure is called sex pili. Now with the help of this sex pili, which is a tubular structure, this bacteria can bind and do sexual reproduction with another bacteria. So say for example, this is another bacteria. Okay, now this bacteria only has its chromosomal DNA and there is no plasmid, so it does not have that F plasmid, hence it is what? F negative or female or recipient. Correct? Now what happens? Through this sex pili, one strand of this, okay, what happens? Let me first zoom it in and show you. Out of these two strands, one strand opens up, okay, and that strands move through this sex pili and goes in the other bacterium. Okay, now once it reaches the another bacterium, what happens that it rolls up to form a circular structure, right? And on this circular structure, a second strand of DNA is formed. Okay, now similarly in the F plus previous one bacterium, this only one strand is left. Again here also next strand is formed on the previous strand, so this also becomes the double stranded. Can you see? Now both the bacteria have the F factor, so even this bacteria, okay, this bacteria has now become what? F positive or male or donor. So after conjugation both becomes F positive, or you can say after conjugation the F negative becomes F positive. Okay, great. One more thing to remember here is during conjugation, can you see the genetic material from this F positive bacterium is transferred to the F negative bacterium? That is, the transfer of genetic material from one bacterium to another bacterium is taking place with the help of, or you can say through, this sex pili. Okay, and this is happening, this process is called conjugation. Okay, this is called conjugation. Do remember this. Great. I hope conjugation is clear to all of you. Now, so we have seen so far three basic methods. First one we have seen in the previous lecture, that was transformation, Griffith's experiment. Then we saw transduction, and finally conjugation. So these three are the three main methods of sexual reproduction in bacteria. Well, when it comes to Griffith, okay, let it be. Well, apart from this, there are some special cases. Okay, this special case is called HFR strain. Okay, this is wrong. It is not written like this. HFR strain. Now, HFR represents high frequency because they reproduce very fast. Okay, or it is also known as sexduction. Okay, so what is it? This is a special case. Let me tell you, it is a special case, HFR strain or sexduction. Now, as the name suggests, sexduction, sex comes for the involvement of sex pili, that is conjugation, and duction because mechanism wise it is similar to transduction. Okay, so it is something like transduction, but it occurs during conjugation. Okay, confusing? Okay, let me clear it out, what it is actually. Now think of, there is this one bacterium, okay, and it is F positive. So this is its chromosomal DNA, and here you have this plasmid. Okay, great. Now what happens many a times that this plasmid gets embedded into the chromosomal DNA. Okay, so let me make it for you. This is chromosomal DNA, and this is the plasmid DNA. Now, plasmid DNA gets incorporated into the chromosomal DNA. Now, if it gets incorporated, then there is a chances that after some time it may get out of the chromosomal DNA and again become a circular independent structure, and this is what happens many a times, and for that it has to be cut precisely at this point, correct? Let me do it in red for clear visibility from this point, but many a times it does not happen. Instead, it gets cut at this point and this point. What will happen? Let me draw it again. So due to that, the chromosomal DNA will appear the same with minor decrease in size, okay, but the plasmid will look something like this. Correct? So this is the plasmid now. Now think of conjugation with another bacteria. So now this particular male bacteria undergo conjugation. Let me do a conjugation. So this bacteria has its own genetic material, and after conjugation, now this also has this strain, but this F factor, the plasmid, is unique as it contains the chromosome from the previous bacteria, right? Then what happens? This conjugation tube is gone. After some time, what happens is again this plasmid gets incorporated into its bacterial genome, and see this white part, which is the chromosomal DNA from previous bacteria, is now part of the chromosome of this blue bacteria, new bacteria. After some time, again what happens is this gets cut, okay, and this time the cutting is at the right place at this point and this point. What will happen? Let me redraw again. There will be this part which has this portion from previous bacteria, and it will get the plasmid alone, right? So can you see during this entire process, the DNA from this bacteria got transferred into the another bacteria, this region, correct? And without use of viruses. This is during conjugation. Hence it is called sexduction, because mechanism wise, can you see, it looks like transduction, but there is no involvement of viruses, rather it is happening through the sex pili, hence it is called sexduction. Such a bacteria shows a high frequency of reproduction, hence it is also known as HFR strain. Great. So what we have seen so far, we have seen three things in this particular lecture. That is, we saw the transduction, right? We saw transduction, then we saw conjugation, and finally HFR strain or sexduction. So this is it for this particular video lecture. What we are going to see next is example of some of the bacteria which are useful, or some pathogenic bacteria. We are going to just look at some examples, okay, and that will cover the eubacteria. Then we are left with the blue-green algae and spirochetes and the other types of prokaryotic organisms such as rickettsia and all. And after that, kingdom Monera will be finished. So for rest of the topics, stay tuned and stay safe. Thank you.